To give you some updates on the work that we've been doing with our ontologies and knowledge graph over the last year, as well as where we're going now. I want to briefly acknowledge all of the team who's been working on this over the past few years, including Ian Braun, who you saw in the last presentation, who's done much of the knowledge graph development, but also Daniel Olson, Emily Hartley, and all of our data managers who are working on this project as well. So Nicole and I are going to, there we go. Nicole and I are going to tag team on this presentation, so I'm going to hand it off to Nicole for, oh, you have your own mic, great. I'm going to let Nicole start by giving you some background on ontologies and knowledge graphs.
Thanks. All right, so how many people here are familiar with ontologies, since we've been raising hands? Okay, I'll give you.
I'm impressed.
Yeah, that's great. I'll give you a bit of a background. So an ontology is a data standard, so there's been a lot of talk these last couple of days about the needs for data standards, and ontology is a type of data standard that's unique in a way because it's represented in a computational language that allows for computational reasoning and inferencing across data. So an ontology represents information in a specific domain or area of knowledge, and what's unique about an ontology compared to other types of standards or controlled vocabularies is that it defines the relationships between concepts in a domain in this computational manner in a way that we can do computational reasoning across the data.
I think the big one, yes.
So an example of an ontology that we work with at CPath is the human phenotype ontology. So this is an ontology that represents abnormal phenotypes represented in human diseases, including common diseases and rare diseases. So in the human phenotype ontology, there's over 16,000 classes representing the top-level class phenotypic abnormality, and then there's subclasses or children classes of this top-level class that are arranged in this hierarchical classification, which are describing different phenotypic abnormalities. So for example, we may have a class like increased urine alpha-ketoglutarate concentration, which is a child of the abnormal class. So all the characteristics of the parent class are inherited by the child class. So this allows for this kind of subsumption reasoning across the data. We also use external ontologies or other ontology concepts to logically define the terms. So like the case of hyperglycemia, we would computationally define that as a type of increased glucose that occurs in an anatomical entity in the blood. So we're working with a group called the Monarch Initiative. That is it's a group that's creating a discovery platform that integrates cross-species disease and phenotype data in a platform. And they've created this gold standard phenotype profile using ontologies. So the gold standard phenotype profile contains over 7,800 rare diseases and phenotypes annotated to these diseases. So these are considered the phenotypes that are typically associated with each of these rare diseases. So these phenotype descriptions are annotated to HPO, or Human Phenotype Ontology terms, and they come from resources including OMIM, the Online Mendelian Inheritance of Man, Orphanet, the Rare Disease Ontology, and Decipher, which includes copy number variant diseases. So we have this corpus of information with phenotypes that are diseases with the phenotypic descriptions associated with them. So we are using this information to build a knowledge graph. So a knowledge graph is a graph database that's a collection of concepts which are represented as nodes in the graph, such as diseases or phenotypes, and edges, which are the relationships between the concepts and the edges, and the nodes and the edges. The edges are the relationships between the concepts. And we can also integrate individual patient data into the knowledge graph, which allows us to query across this data and derive new information and answer questions about this data. So we're hoping that we can add value to individual patients and people living with rare diseases by integrating this patient data into a knowledge graph and enabling querying across this data. So we also are building the CPath ontology, or the critical path ontology, which is an application ontology that's integrated into our knowledge graph. And this is an application ontology that's built for the specific purpose of driving our knowledge graph. And it reuses many terms from existing ontologies to represent types of concepts in this domain. So we have information about diseases, phenotypes, laboratory measurements, and functional tests, so we can standardize the data that we're gathering in the RDCA-DAP and be able to standardize it using this ontology. And this is an open source ontology, and it's available at the URL listed here.
Thanks. I need the clicker. Thanks. Okay, so now that you're all experts on ontologies and knowledge graphs, or at least have some basic understanding of them, you're probably wondering what we're doing with them in the RDCA-DAP. So that's what this next section is about. The key thing that we're doing is building our own knowledge graph. And what this knowledge graph does that is different than many of the existing ones that are out there already is that it integrates the individual patient level data that we have access to through CPATH, through our consortia and RDCA-DAP. That's a really unique aspect of it. We're integrating that data with the existing data from other knowledge graphs like the Monarch knowledge graph that provide a real wealth of information about phenotypes, genes, diseases, a number of sort of known facts and associations, but all at the population or genotype level. So by bringing together this wealth of knowledge that already exists at the population and genotype level with our individual patient level data, we're able to do things like enhancing data integration and discovery across diseases, identifying cohorts or subsets of patients within heterogeneous diseases, translating back and forth across standards for different use cases, supporting diagnoses, and suggesting possible outcome measures for diseases that lack one. This is a screenshot of the portal where you can go to where one can go to query the knowledge graph. We have not yet made it public because it does contain patient-level data. We're working on ways to set up access permissions so that users of the RDCA-DAP portal will also be able to go in and do those queries, sort of the same way you do with the cohort builder that Kara showed, where you can do queries and get generalized results without seeing the patient level or line level data at one point. This screenshot, I'm sorry, the text is really quite small here, but this shows the number of annotations that are, or associations that are currently included within the data, within the knowledge graph. We've only got a few thousand patients in there at this point. They're coming from a rare cancer desmoid tumor from the DTRF, rare epilepsies from the Candon and Ren patient organizations, as well as some of the data set from the Friedreich's ataxia integrated database that you saw earlier today. There's also a synthetic data set, the Synthea data set that's widely used that you can use to do some exploratory analysis in the knowledge graph. And so even I can't read those tiny little legend on this figure, I realize now, but this just shows you that there are different associations in the graph between patients and drugs, people and diseases, people and the one all the way on the right, the sort of light green bar, aqua bar, is the associations between patients and phenotypes. And you can see that's the data that we have the most of because those are the associations that we annotated and created through our own data sets. Because the knowledge graph is built on an ontology that has a hierarchical structure, and we're using the same ontology across multiple data sets, we can start to aggregate and do queries across data sets. So what this graph is showing you is that looking at all of the data sets, that is the cancer, the epilepsies, and the Friedreich's ataxia data. Again, apologies for the very small text on the margin here, but these are showing high-level anatomical systems from the anatomy ontology. And if you look at all of our data sets, you can see that most of our patients have phenotypes that are associated with the nervous system. There are also a number of patients that have phenotypes associated with the respiratory system and some within the digestive or gastrointestinal systems as well. Now, if we break this down into individual diseases, we could look, for example, at the tumor patients from the DTRF data set. And you'll see that most of the patients have phenotypes that are only associated with the respiratory system. It's a cancer that occurs, tumors that occur within the lungs. There are also a number of phenotypes associated, again, with the digestive or gastrointestinal system. In contrast, if we look at the Friedreich's ataxia data, these are phenotypes taken from the mFARS rating scale. Most of the patients have phenotypes that are associated with the respiratory system, with the nervous system, not surprising that's given that it's a neurological disease. So having data aggregated at this level, this is a very high level example, but as we get more data and we'll be able to do much more complex queries. And just to point out, these are charts that were generated on the fly given a query, saying, show me this, and this is what it spits out. Thanks to Ian, who can write the wonderful code to do things like that. And so, you can say, look across all of the data within RDCA-DAP, find me all of the patients, regardless of where their data came from, who have an abnormality of their limbs, have a muscular disability, any kind of phenotype that you want to query across at all different levels of organization. And again, because of the hierarchical nature, it will aggregate. So nervous system is including all of those phenotypes that have anything to do with the nervous system, but you can drill down and get more specific if you want to. So before I go into, and Nicole and I go into some of the specific examples of analyses that we're doing with this knowledge graph right now, I want to talk a little bit about a technique that we're using called semantic similarity analysis. Semantic similarity analysis uses the knowledge that's encoded within ontology, that sort of node and edge relationship, and the semantic reasoning or computerized reasoning that you can do with those graph databases to create a matrix of similarities among all of a certain type of entity within your graph. Now, that could be between all of the diseases, it could be among phenotypes, it could be among people. On the example that I've got here on the right, I'm showing an example of semantic similarity analysis on anatomical entities between the renal glomulurus and the glomerular capsule. Now, renal glomulurus is inside a glomerular capsule, and there are many different ways that you can calculate semantic similarity, but essentially they all use this concept of how much of that graph, the paths within that graph, did the two entities share in common. And if you look at, again, small writing, boy, the two different terms, look in the middle, those are all of the terms and the nodes within the graph that they share. And so, not surprising, they share a lot of nodes, they have a lot in common, so they have a very high semantic similarity. If I were to do something like compare renal glomulurus with alveoli, which is kind of a similar structure but in a very different part of the body, there would still be some overlap, but less of it, and so they would have a lower semantic similarity. So what we've done within our knowledge graph is we've taken all of the phenotypic profiles, that is, for every patient we've recorded all of the phenotypes that we know about them, not just a single one. We've put those into the semantic similarity analysis, and we calculated a large matrix of similarities among all the patients. So how similar is, if Nicole and I were patients, how similar is my phenotype to Nicole's phenotype? Pairwise by every possible set of populations within there. Then with that data, we can do all kinds of different things. And so we're going to give you a few examples of the things that we're doing with them. This is to present a high-level view of one of the things that you can do, which is just visualizing the data. The matrix, of course, because it has many phenotypes, is a multidimensional matrix. But there's a tool that I'll talk about in a few slides that can compress that matrix down into 2 dimensions for visualization, similar to like a PCA, except it's using very different methods, and then cluster those results for visualization. So you can see here based on the parameters and how many clusters, et cetera, this is clustering all of the data. This includes the synthetic Syntheia data set. That's why there's many different points. That's the cloud on the left. So if I click here, it will show you the exact same set of data points, but color-coded by diseases. It's a little hard to tell, but all the way on the right side, there's a slightly bluer green. Those are the Friedreich's ataxia patients. The dark blue near the bottom middle are the desmoid tumor patients. That yellow little oval, not a rectangle, is our rare epilepsies, and then the larger green cloud is all of the Syntheia. This is not showing their clusters, it's just a sort of manual color code, not manual, a scripted color coding of the diseases, but you can actually use this to see that the clusters correspond to the different disease data sets to a large extent within what we've got. Okay, so diving into a little bit more specific detail, I'm going to look at some of the Friedreich's ataxia data and the work that we've used this knowledge graph to do, some of the initial analyses. So the modified Friedreich's ataxia rating scale, I don't think I need to go into a lot of detail here because we had some great presentations on this yesterday that talk about what mFARS is. But one of the key things I want to point out is that as we were mapping the Friedreich's ataxia rating scale to ontologies, First of all, this was a proof of concept to just even show that we could do this. So taking something that's a question and answer pair and putting it into ontology is not a trivial task. So yes, we showed, but we could do this, and we could come up with things that make sense. We took each question and associated it with the biological trait that it was trying to get at. Was it motor function? Was it speed, et cetera, and also the anatomical entity that it was associated with, that a limb, the face, the gastrointestinal tract, et cetera. So we've got that data. The steps that we took to do this analysis were that we first evaluated the whole COA instrument. We modeled the instrument components and questions to ontologies. We used many of the terms that we needed to describe them were not in existing ontologies, so we worked with those ontology developers to create new terms where they were needed. We ingested all of the data and all of the ontologies into our knowledge graph, and then we did queries across that knowledge graph. And one of the pieces that we got out of it was a semantic similarity matrix that I showed you a minute ago. And here's that same matrix, focusing just in on the clusters that for FA. These are the same, they look a little bit different in this graph because they've been pulled up, but it is the same exact clustering approach that was used for all the data. We didn't re-cluster just within the FA. It's from all of the clusters. You can see, as you might have noticed in the other graph, there were two clusters within Friedreich's ataxia. This is the method, the T-sign distributed stochastic neighbor embedding is the method that does the clustering, and then the dimensionality reduction. And Ian can yell at me if am I getting that right? Does T-sign also do the dimensionality reduction? Thank you. K means clusters, and the T-sign just does the reduction of dimensions then. First I saw this, wow, this is really exciting. We've got two really distinct clusters of FA patients. What's going on here? Well, it turns out that, cluster 1, the blue cluster, is an individual data set. This does not include the FA-COMS data set within here. So it includes the other 5 data sets that we have available for Friedreich's ataxia right now. And it only includes the patients that had data on the particular, on mFARS within there. And it would be cool. So it turns out that cluster 0, you can also get, it will also report on you which ontology terms are enriched within each of those clusters. And it's kind of cool because cluster 1 has a lot more enrichment for facial abnormality phenotypes, whereas cluster 2 has a few facial phenotypes enriched, but more along upper, like limb abnormalities. But we don't actually think that's a big biological difference. We think it's just what one data set collected versus what the other data set collected. And to me, this is so, like, it's confirmatory. This is what we wanted. We didn't expect really earth-shattering results with this initial analysis because FA was fairly well known. We actually wanted to use a data set where we could have some predictions about what was going to happen and see if they were confirming those. So they just asked different questions in one survey. But the nice thing about this is when you're trying to look at a data set for initial analysis, it gives you another way to visualize it and say, where might I have problems, right? Do I want to include this data set, number 6? Because maybe it's different than all the other ones, and this is a great way to look at that. All right, I'm going to hand it off now to Nicole to talk about some of the work we've been doing with rare epilepsies.
All right, so we have some data coming from 2 data sets from rare epilepsy groups. And we did an analysis to try to look at the semantic similarity across phenotypes for these patients. And we wanted to kind of do this proof of concept where we had done some research like this initially. This is work that was done by the Monarch Initiative. So I'm just going to give you an example where we know that we can actually use the semantic similarity metric to diagnose patients with rare diseases, and then show you some examples of work we've done at CPAT to try to utilize some of the individual patient data that we have to compare phenotypes from the gold standard phenotypes to our actual individual patient data. So this example here, like I said, is work done by the Monarch Initiative, where 2 patients came to the clinic and they presented with mutations in the same gene, but they had varying phenotypes, and these patients were undiagnosed at the time that they were seen in the clinic. And these patients had a collection of phenotypes that are described on each side in green and in orange. And these patients didn't match up any typically known disease. But looking at the gold standard phenotypes for this Wiedemann-Steiner syndrome in the middle in blue, these are the typical phenotypes associated with this disease. And these patients didn't match these phenotypes exactly, but some of them had some fuzzy matches or some similar overlapping phenotypes with the gold standard. Like, for example, the patient on the right, the boy, had a long toe, where the typical phenotypic presentation is a short toe, but these are both abnormalities of the toe. So if you were looking at the hierarchy, you could see that these are in the same branch of the ontology. So using this phenotypic similarity metric, the physician was able to diagnose these patients with actually having the same disease, even though they didn't have the typical phenotypic profile. So we have this individual patient data at CPath coming from two individual data sets, one the CAND data set, which is the KIF1A associated neurological disease, and a set of patients from patient registry from the Rare Epilepsy Network. So these surveys describe patient diagnoses and their accompanying phenotypic profiles. So we leverage the Monarch Initiative Knowledge Graph and gold standard disease to phenotype annotations and performed semantic similarity analysis against our individual patient data. So our individual patient data was mapped to these ontology terms. We annotated to the ontology terms. This is work done by Daniel Olson in our group. And then Emily Hartley, one of our data engineers, performed this analysis to first compare if the patients in the CAND data set, the CAND disease is a group of disorders which include some common diseases like autism and epilepsy, and also some monogenic diseases, which are described here on the bottom of this graph. So we looked at the phenotypic presentations in the patients from our data sets and compared them to the phenotypic presentations that are typically associated with the patients from the gold standard data set that we had. So the numbers on the bottom correspond to the percent of similarity between the patient data and the gold standard data. So if we look at the center example, the intellectual disability autosomal dominant 9, we can see that there's dozens of patients that have between 50 to 60 and 60 to 70% similarity to the phenotypes that are typically associated with this disease. And if we look at the other two diseases, we see that these patients don't have a lot of phenotypic overlap with these diseases, suggesting that these are probably not the diagnoses that these patients have. Also, looking at the REN data set that we had patients who had reported that they had known diagnoses or had been diagnosed with a certain disease, which are listed on the x-axis of this graph. And we compared the phenotypic profiles from the gold standard that are typically associated with these diseases, listed on the x-axis, and compared the number of patients who matched the phenotypic profiles in the individual patient data sets to the gold standard. So like looking at a Cardi syndrome all the way on the left or going towards the center, the LGS, the Lennox-Gastaut syndrome, or West syndrome on the right, we can see that there's, yet again, dozens of patients who have over 60% or up to maybe 90% overlap with the phenotypes that are seen in these profiles, whereas as a lot of the other patients, and there's also a lot of patients in these particular diseases who have differing phenotypes from what we see in the gold standard. We're also able to view this data using a heat map as well, using this method called PhenoGrid. So here we have an example where on the x-axis on the top, this corresponds to each individual patient, and this is for one particular disease, tuberous sclerosis complex. And so on the y-axis on the left, these are all the phenotypes that are associated with this disease. And then the darker colors correspond to high semantic similarity between the two phenotypes in the patient data compared to the gold standard. And the lighter colors, the green boxes, indicate lower semantic similarity. So if we look at the first patient, we can see that they have a lot of similar overlapping phenotypes to the typical phenotype seen in this profile. Some of the blank spaces indicate they don't have that phenotype at all. So, and then if you look at the darker colors, like on the right, the infantile spasms, this has a 98% similarity, where the patient reported they had myoclonic seizure, but the gold standard says they have infantile spasms. That's because these are basically siblings in the same branch of the same hierarchy. Whereas at the bottom, the shagreen patch, the gold standard, this is the gold standard phenotype that's seen in these patients, and one of the patients reported they had dental enamel pits, but this is only 36% similar because they're both, if you go up the hierarchy, they're both types of abnormality of the connective tissue, but they're in different branches of that hierarchy, so they're actually not very similar, but they do have kind of a high up common ancestor. So it was really interesting to see these results, and we definitely have more querying and analysis we want to do with this data, but our initial takeaways from this are, one, there could be gaps in knowledge, and this gold standard data set is what we're working with, but there could be really other gold standard phenotypes that are associated with these diseases. So we would like to look into that further. We also took this from patient registry data, and it's really hard to determine if the survey questions in the registry data cover all the phenotypes that are in the gold standard. So that is something that we also plan to follow up on and look and see if the registry is covering all the phenotypes that we are intending to examine. And another thing about the registry data is that it's not also meant to capture the natural history of the disease. It's meant to capture the disease status at a certain point in time. And it's also kind of on a rating scale. So it's not always capturing the exact phenotypes that are representing the disease in itself. It's more representing the patient's reporting at that time point. And there's also a lot of phenotypic noise. So patients may have a rare disease and present with certain phenotypes, other co-occurring disease that may present with certain phenotypes, and that can be reported on the registry data, which may be irrelevant to the actual rare disease that they have. And then also there's been difficulty with the mapping, doing the mappings themselves. The survey questions can be very vague and sometimes hard to interpret, so there's a lot of subjectivity in doing the mappings themselves.
Thanks. All right, so just to wrap up where we're going to go with this and you know, what we expect to do in over the next year or so, we're going to addressing the semantic similarity issues that Nicole just outlined in the previous slide. We want to do different activities to identify and fill the gaps in the rare disease phenotypes within the natural history. That could include analyzing real-world data and then contributing that knowledge back to open open phenotype descriptions like the Monarch graph. We want to work with communities to standardize data. I'm really excited yesterday to hear several people recommending that we start to standardize surveys and COA questions. We'd love to give input on how you can use ontologies to do that. We definitely want to reduce the noise that we're getting. One way to do that would be to provide a good mechanism for different individuals, whether they be patient groups or industry or even individual practitioners, to contribute to gold standard phenotype profiles when they know them. We want to improve our mapping efficiencies. There's a lot of work to do here. You know, in terms of the COA analysis, like I talked about, that the FA data was a proof of concept for. There's a number of ways that we would like to build off of that. We are currently mapping 4 different instruments to the CP-RND ontology, and of course we're also reviewing all of those wonderful resources that Lindsay talked about in the RDCA-COA resource yesterday, the COAs that are there. We want to do additional semantic similarity analyses that include phenotypic descriptions from existing COAs, but also possibly include additional types of phenotypes from labs or imaging. And we would love to incorporate longitudinality. The FA data that I showed just treated each time point as an individual point, that we get the same results when we cluster by patients. Long term, we would love to have a sort of a COA recommender. We've had a number of conversations with Lindsay about this and how we can make this happen. You know, that would help you, that would build on top of the resource that's already there from the RDCA-COAC that's a real mouthful, isn't it? But help you select you know, based on the phenotypic profile of the disease that you're interested in, would help you identify other COAs that might be useful. We think that there's a lot of potential utility. We've been doing this now for about 2 years, and it's been kind of slow going because there's a lot of we're doing things that are really new. There's a lot of work in ontologies out there, but nobody's been doing it quite the way that we've been doing it. So we spent a lot of time just figuring out how to do this, and I feel like we're really starting to get up to speed. And I hope that the work we've been doing can be of great use to the rare disease community going forward. We'd love to get your input and, you know, potential ideas about how you might want to use this within the groups that you're working with. Some of the key uses for ontologies include data harmonization, integration, and discovery. I showed a very high-level example of how you can use ontologies to search across diseases and data sets that would normally be isolated or siloed. Nicole gave a great example of how ontologies and knowledge graphs can support diagnoses. You can imagine using that kind of similarity and fuzzy matching to go into EHR records and search for patients who might have a disease that have not been diagnosed yet. The example that I gave, even though there were not, as expected, there were not really great, interesting cohorts or subsets of patients within FA, we are very confident that we'll be able to use this to identify subsets of patients within rare diseases. And then it can even do things like using, when you're looking at inclusion-exclusion criteria, rather than using a single criterion, you can use the semantic similarity or the complexity of the knowledge graph to look at patients as a whole set of their phenotypes, not just as which disease they have. And finally, the COA recommender that I mentioned before is something that we would love to see. That's something that we're going to be spending a lot of time in the next year. And I think we're about out of time. But if there's time, we can take questions, or if not, if we're at the end of the session, I'm happy to stick around during the break for any questions that people have.
We can take a few questions.
Okay, so we've got time for a few questions.
Hi, thanks for that interesting presentation. Doug Langman, University of Iowa. I was wondering, with your gold standard phenotypes and semantic measurements of semantic similarity, is there any weighting involved in terms of the importance of various features of a phenotype? I mean, many diseases have certain features that are necessary or possibly sufficient, or we can talk about that in terms of sensitivity and specificity of different features of a disease. Is there any attempt to?
I think, Nicole, you can answer that best.
Yeah, so the gold standard phenotype annotations from the Monarch Initiative actually include frequency information, like how frequent the phenotypes occur in the patient data or in the patient populations. However, in the analysis that we did, there was no weighting for the frequency. So that is something else that we would like to take into consideration in our future analyses.
Okay, thank you.
My question, and I don't know if this de-identifies, I loved the semantic, the diagnosis, I loved all that. And one thing that I was thinking is behavior may play a role, because how I acted when I was growing up in Texas is not how I acted in Boston, and it's not just because I'm older. So can you break this down and say, let's take a cluster of people in certain geographies and see if this is similar, because I wonder if my behaviors, hanging out outside versus staying indoors, coats and stuff like that, is also going to modify the outcomes that you're seeing.
That's a great point. Yeah, that's something that we haven't considered really, but it would be lovely to include that into the analysis. And with the knowledge graph, we have the ability to incorporate other kinds of modalities too, so that's something we should definitely consider.
So there's a couple of challenges there. I mean, one is, do we have the data to do that? The second is, do we have the ontologies? And so I've done a little bit of work on ontologies for social determinants of health. The environmental health community is particularly interested in that. But I would say that Nicole and I have both been very active over the years in the Open Biological and Biomedicals Ontology Foundry, or OBO Foundry, where many of the well-known ontologies like the human phenotype ontology, the gene ontology, the Uberon anatomy ontology sit. Though there are not, those are a great set of ontologies to work with, there's not a lot on social determinants of health, on behavior, on environment. There's a wonderful environment ontology, but it doesn't describe the kinds of environments that you're talking about. So before we can make progress, like what we're doing now is taking advantage of the ontologies and the kind of knowledge that already exists. It will be another 5 years before we can start to add in those other data sets, unless somebody wants to give us $1 billion, then we can build the ontologies.
The knowledge graph and ontologies are open too, so you're welcome to do the studies yourself too.
I have a quick question, almost to dovetail from the conversation that was just started earlier. It's like, if you could apply this to ALS, your ontology, we have all this omics data, we have, income data, we have natural history studies. How would what would you be able to do with the ontologies work that you're doing?
Well, I think that one of the ALS would be genomic diseases are great, especially when when the variants are known, because there is so much information on how different genotypes are related to proteins, related to pathways, related to expression. There's a lot of information out there, so you can kind of plug your gene variants into the existing Monarch graph get the information that, and Nicole, you can like slap me if I'm misrepresenting, but this might, you can get you can use the Monarch and other sources to have access to that wealth of omics data that's out there and combine that with individual patient data. That's where we would love to go with this eventually. Thanks. Oh, yeah.
Yeah, hi. I just, Jackie French from NYU. I just wanted to add to the gentleman from Iowa in regards to weighting, and that is that, for example, in the rare epilepsies, almost every rare epilepsy has like 5 things in common. They have developmental delay, they have autism, they have, multiple seizure types. I could go on and on. Sleep problems, behavior problems. So it's really, when you look at those graphs, you're going to see lots of things that are the same, but can you sort of sort out the things that are actually different so that you don't just say, oh, all these things are the same?
So I think that really addresses the fundamental issue that Nicole, as we were preparing this talk, something we were really aware of was that the way the data are collected, the purpose for which they're collected is one thing, right? So if it was, for example, the mFARS rating scale, that was designed and validated specifically to understand where someone is in a progression of Friedreich's ataxia. It's not designed to describe the full phenotype of every person who has FA. Likewise with epilepsies, right? The registries are collecting data for a particular purpose to understand what may be important. They're not collecting data for the gold standard of describing that phenotype. But I've heard over and over again over the past 2 days how important it is to understand the natural history of all these rare diseases. So maybe we need to look to the people who are collecting data and say, can you start gathering, or can we minimize the burden on patient and start getting that information from real world data? Are there other ways that we can start to really understand the natural history of all of these rare diseases through the ontologies?
And combining it with genetic information too would also be really valuable if we had information about the genotypes of genes affected in different variations. That would also be relevant as well.
Yeah.
Just a comment for everybody. I'm reasonably familiar with the ontology field. I can tell you Ramona is the single only person in the entire world who can excite an audience around ontology.
Sorry, Nicole. Nicole's pretty good at it too.